Today our topic is on sphingolipids. Okay, so normally the lipids, it is also one of the class of lipids, but generally in the structure of lipids, we'll have the backbone of glycerol along with the fatty acid chains. Whereas instead of glycerol, in place of glycerol, we have a long aliphatic amino alcohol that is known as a sphingosine. So instead of glycerol as a backbone, we have a sphingosine as a backbone. The general here the chemically it is known as chemically a palmityl CoA. The amine group amino acid which is attached here is a serine group. Serine amino acid is attached. And the fatty acid is the fatty acid long chain. The tail is attached to the polar head at the second carbon. The amide amino group is linkage is known as the amide linkage, which forms a ceramide structural parameter. Okay, so this is just a difference between your normal lipid structure and the sphingolipids. So generally, the lipid will have the polar head, that is your head will have the glycerol as the backbone, and the tail will be a fatty acid chain. Here, the sphingosine will be your polar head, whereas the hydrophilic tail is your fatty acid. Okay, so we have different classes of sphingolipids: phosphosphingoside, glycosphingoside, and sulfatides. So from that, phosphosphingoside, it is in biological membrane as a sphingomyelin. So the sphingomyelin it is known as a sphingomyelin biologically in the membrane as a sphingomyelin. So only sphingolipid with a phosphate group and no sugar unit is attached here. So you have a phosphate group attached over here. So nitrogenous group is choline. So choline amino acid is attached. So which is an important compound of your myelin of nerve fibers. So the myelin which is present in the nerve fibers, it is because it consists a component called phosphosphingosides. So the sphingolipids or glycolipids, which is also known as cerebrolipids, which occurs on the outer face of your plasma membrane. Plasma membrane, which consists of the outer surface that contains your glycolipids or cerebrolipids. So which do not contain phosphate groups. So generally, we have a phosphate group attached to the earlier, which we saw that sphingosides, which contains a phosphate group. To that is your amino group is attached. So here there won't be any phosphate group attached to it. So one or more sugar unit is attached to the hydroxyl group of carbon 1 ceramide. Then it is known as cerebroside. So it is mainly seen in the brain. So it is called as a cerebroside. So in the brain which is present, the glycolipids which is present is known as a globosides. Okay, galactocerebroside seen in cell membrane of your nerve tissues. So this type of glycosphingolipids will be present in your nerve tissues, or it is known as a gangliosides. Glucocerebroside is a non-neural tissues which is present. So glucobioside with 2 or more sugar units. So usually we have D-glucose, D-galactose, and N-acetyl D-glucosamine will be attached to the, sphingo backbones. Okay, so sulfatides, gangliosides, so which is known as the most complex of the glycosphingolipids, with oligosaccharide as a polar head. So here the oligosaccharide is will be as a polar head. So we have a sphingosine as a polar head, whereas in ganglioside the polar head will be a oligosaccharide. Okay, so presence of sialic acid, which is known as a N-acetyluraminic acid, a sialic acid. Okay, which gives your negative charge at neutral pH. So it is called as acidic glycosphingolipids. So you have different types of gangliosides, GM1, GM2, etc. NGD, TGT, TGQ series, so which differs depending upon the tissues. So the role in cell recognition and tissue differentiations. The main role of the gangliosides is to recognize the cell and helps in the differentiation of the tissue. The differentiation of the tissue means it gets multiplied or it gets regenerated to the next form. So that is mainly maintained by your gangliocytes. So in case of to define your human blood groups, so in that case also your sphingolipids is helpful. So generally the O antigen contains the sphingosine backbones and a fatty acid chain. Along with you have a oligosaccharide attached. Okay, glucose, uh, galactose N-acetyl neuramic acid, galactose, and okay. So you have a ceramide head and a tail with a so that it which is the sphingosine that your sphingosine is attached to a sugar groups. So that is the O antigens, which differs in your A antigens and the B antigens. You will have a see here in O antigens you have one GalNAc, whereas in A antigen there is the another GalNAc it is attached, whereas in B antigens you have only galactose, not the GalNAc. That is neuramic acid is not attached. It is not in the form of neuramic acid. Okay, that is sialic acid form. So this helps in the differentiation of O antigen and A antigen and B antigens. So which if the ganglioside is stored. So normally I have told you that glucose when it is stored in your in the liver, glycogen disorders, glycogen storage disorders happens. In lipids also, if the lipids are that is lipids will be stored in the form of triglycerides, which leads to fatty livers. Similar way, when the gangliosides are stored in your system, it also causes some disease, the storage disease. This is known as genetic defects in your lysosomal enzymes to degrade ganglios, which is because of there is a defect in the lysosomal enzyme production in your systems. So if there is any genetic deficiency that which leads to the production of lysosomal enzymes, so these are the enzymes which helps in the degradation of gangliosides. Okay, so that is because of ganglioside storage disease and Niemann-Pick disease. Niemann-Pick disease is because of sphingomyelinase, lack of sphingomyelinase enzymes. The similar way, Tay-Sachs disease is also due to the lack of hexosaminidase A enzyme. So this is the normal sphingolipids metabolic process and the turnover. Okay, so here if there is any if there is no hexosaminidase enzyme production here, the gangliosides GM1 will not be GM2 will not be converted to GM3. Okay, so then the GM2 form itself is stored. So hexaminidase A is missing. So which leads to the Tay-Sachs disease. So here the similar way, if there is a deficiency of hexaminidase A and B, which is the turnover conversion of globosides into exosylceramide. So that leads to Sandhoff disease. Okay. So the similar way, alpha galactosidase enzyme. So if there is a deficiency of production of alpha galactosidase A enzyme, then it leads to the Fabry's disease. Okay, so each enzyme plays a main role in the conversion of metabolic process. If these metabolic process is not properly carried out, then it leads to the disease condition. So each enzyme has a specific function. So each enzymes are the proteins and each proteins are the genetic code which helps in the which is we target for the disease conditions. Okay, so the particular gene which is responsible for that specific enzymes production. So if there is a genetic deficiency of this particular enzyme, then it leads to the so sphingolipids must turn over to prevent the accumulation in the neuronal cells. If that if, if it is stored, then it leads to the formation of Tay-Sachs disease. The other form of sphingolipids, that is sulfatides, sulfate group will be attached to your ceramide oligosaccharides. So highly multifunctional glycolipids is a form of sulfatides. So the main function of your sphingolipids. So first function is cell signaling mechanism. So particularly in the form of ceramides, it plays a main significant role in the regulating of your cell growth. So cell growth differentiations are mainly carried out by your ceramides and apoptosis. That is your programmed cell death. That is also that that signaling mechanisms are triggered by the ceramide component. The membrane structure and fluidity. So mainly your membranes, the cell membranes or the nervous systems of outer membranes are uh, the flexibility or the because of the structural integrity is mainly based on the sphingomyelin compound. A type of sphingolipids, which is a major component of your cell membrane, which helps in the uh, integrity of the structure, particularly in the nervous system. And cell-to-cell recognition. If these cells-to-cell recognition occurs only, the other signaling mechanism will carry out. So glycosphingolipids is such a, for example, a cerebrocyte and gangliosides are one of the main sphingolipids which is involved in the cell-to-cell recognition and adhesion process. So that is, the cell has to bind to the specific receptor. So one cell will give an information it is like a messenger. Okay, so you're sending a message to one person, and that person is the mediator of your information. Okay, so he has to send that. So similar way, for example, if I'm informing Kirtika, Kirtika will inform the messages to the classes, right? This, the, this is because that I recognize that she is the representative, that she only can carry the information to the side. Okay, the same way here also the information will be carried on by through these mechanisms. Only these cells can recognize the functioning process. So that is why they play a main role into the cell-to-cell recognitions. Particularly gangliosides, which is involved in the neuronal tissues and it is also uh, plays a main role in the brain functioning process. So brain mainly your information which is taken to your brain through your uh, signaling mechanism, through your eyesight, through your hearing knowledge, through your mouth or anything. So information has to pass to your brain, that you have to do this. So that is all done by your gangliosides. Gangliosides. So they are mainly involved in your uh, brain function. So next is lipid metabolism and transport. So I told you that if any, so all the biomolecules are involved in your uh, body metabolism. So every mechanism, so all the mechanism which is in your body, we work, right? The similar way in your system, all the metabolism has to be carried out. So each metabolisms are carried out by your enzymes. So whatever may be, you are eating or you are sitting or you are doing something, everything for every uh, movement of your systems, your some cells are behind which is working day to do your work. Okay, so if that cells triggered out or the cells are dead, that leads to some dysfunction. That is due to some of the dysfunctioning, right? So the enzymes are the main component which is involved in the conversion of the mechanism of each metabolism. They are the background which is involved in the conversion of one component to the other component of metabolism. So sphingolipid metabolism is one such important metabolism and transport mechanism which is involved in your system, such as sphingomyelinase, ceramidase are the main enzymes which regulates your turnover of sphingolipids, which she influence to balance that is to have a homeostasis mechanism and to help the lipid related disease. Okay, and next is your cellular stress response. So all the stress which is generated in your systems. That is because of our outer external sense or internal sense. Ceramides is a particular one which is involved in the generation of the response towards your various stresses. So whatever the stress which is occurring in your system, it is responded by your sphingolipids, particularly by ceramide. It acts as a second messenger to trigger the cellular stress responses. It may be a apoptosis mechanism, it may be autophagic mechanisms. So whatever. So depending upon your cellular condition, depending upon your stress condition, the response may be varied. Next is main. It is on the next mechanism function of your sphingolipids is it is involved in the regulation of immune response. So there are various types of sphingolipids. It's one particular type is ceramides and sphingosine 1 phosphate and other sphingolipids which is involved in the regulation of your immune cells. That is particularly your T cells and B cells and macrophage. So your T cells and B cells are the main immune mechanism involving component, and the macrophages. They are the player of your immune systems. Thereby, they influence your immune system function. Okay. Then the pathological implications. If there is any dysregulations in your sphingolipid metabolism, uh, previously I told that if there is any deficiency in the enzymes, so that is due to genetic defects. These pathological implication is because of disorders or disease conditions, various disease conditions. It is maybe due to this dysregulation of the sphingolipids, maybe due to the cancer condition or due to the neurodegenerative disorders or many other metabolic disorders. Carbonic metabolism cycle. It may be a carbohydrate metabolism. It may be a protein metabolism. Something. So any other metabolic disorders or any other inflammatory condition may also lead to the dysregulation of sphingolipids. Okay, these are the pathological conditions. These are the main biological function of your